Good afternoon, and welcome to the fourth section of today's symposium. My name is Richard Davey. I'm the Deputy Clinical Director of NIAID, and it's my privilege to moderate a session in which we'll have three distinguished speakers talking about themes that Dr. Fauci touched on during his initial talk. The first will be the overview of vaccine candidates by Dr. Peter Hotez. The second will be a review by Julie Ledgerwood of the current state of vaccine clinical trials. And the third session will be on ongoing therapeutic trials and treatment guidelines by Dr. Clifford Lane. We'll start with Dr. Hotez. There's one caveat, however. Dr. Hotez had a conflict, so he was not able to attend this session in person, so he sent a pre-recorded talk for his session. For those who don't know, Dr. Hotez is Dean of the National School of Tropical Medicine at Baylor. He's also a professor in the Department of Pediatrics and Molecular Virology and Microbiology. He holds an endowed chair in tropical pediatrics, and he is co-director of the Texas Children's Hospital Center for Vaccine Development. So with that, why don't we run Dr. Hotez's pre-recorded lecture.
Thanks so much for the opportunity to speak to you today about COVID-19 vaccines with my twist in a time of anti-science. I'm a vaccine scientist and a pediatrician, and I co-direct the Texas Children's Center for Vaccine Development, and at Baylor, I'm also a professor at Baylor College of Medicine, where I'm also dean of our National School of Tropical Medicine. We've been looking at some of the forces that have been driving back disease, particularly over the last 5 years. So we've noticed actually a decline in all of the great global health successes that we've had since the year 2000, since the formation of the Gavi Alliance, the Global Alliance for Vaccines and Immunization, in partnership with WHO and UNICEF and other vaccine partners, and have noticed that there's been a slowing, halting, or even a reversal of gains. And it's happening in very interesting and unusual ways. And it's happening in parts of the world where I've listed there in white there, where political instability combines with climate change, poverty, urbanization, deforestation, or anti-science. So a lot of social determinants together with climate change. And I have a new book that will come out next year called Preventing the Next Pandemic, Vaccine Diplomacy in a Time of Anti-Science. And as part of that, with a group at Texas A&M, we've been looking at all of these social determinants to plot a vaccine risk index. So many of you may know the work of Professor Heidi Larson at the London School of Hygiene and Tropical Medicine. She's looked at a vaccine confidence index, and we've now glommed onto that some of these other big picture issues like political instability, poverty, climate change, urbanization, to create this risk map where we're concerned we're going to see the return of vaccine preventable diseases. And the point here is that we started this analysis before COVID-19 came on the landscape, so that COVID-19 is actually the culmination of some downturns in our ability to vaccinate the world's population. Rather than something totally game-changing. Now, specifically with COVID-19, what we've noticed among the 30 million cases and 1 million deaths is that overwhelmingly they're occurring in G20 countries, group of 20 nations, like the United States, Brazil, India, Russia, South Africa, Mexico, and Saudi Arabia. And predominantly it's the poor living in these G20 nations. And this is very much in line with something that I've been writing and speaking about for the last few years, this concept of blue marble health, that essentially says when you look at global health problems, rather than developing versus developed countries, in fact, what we see is that the G20 economies, the 20 wealthiest economies, actually account for most of the world's poverty-related diseases. Now you might say, well, that doesn't make a lot of sense. Well, except when we look at WHO or Institute for Health Metrics data, it's the poor living among the wealthy that account for most of the world's poverty-related diseases. And I gave it a new name, Blue Marble Health, to differentiate it from traditional norms of developed versus developing countries. For full disclosure, it's an idea that is not really caught on yet, but we've now done an update around this with COVID-19, and you could download it for free through Johns Hopkins University Press Project Muse, and it's called Poverty and the Impact of COVID-19, the Blue Marble Health Approach. And give you an example of that, here in Houston, where I live and work, the Houston Health Department puts out the people who've perished in our COVID-19 epidemic on a daily basis. This is from earlier this month, and they don't provide names, but they'll provide sex, age, and race or ethnicity. And this is what we're seeing every day. Hispanic, 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 Black, Black, Hispanic, Hispanic. It's predominantly people of color, people living in low-income neighborhoods, especially the essential workers, those who can't make a living via Skype and Zoom and are working at construction sites or family-owned businesses or in the transportation sector. They're the ones really getting hammered in this epidemic, and it very much goes by the Blue Marble Health concept. Now, in terms of shaping vaccines, I like showing this slide from Dr. Fauci, where he showed it earlier this summer at the National Academies. And it basically points out that, when you think about COVID-19 vaccines, even though there's a wider range of technologies, they actually more or less work by the same principle. They're all focused on inducing a strong immune response against the spike protein of the virus. And by now everyone has seen cartoons of the SARS coronavirus type 2 and the spikes that allow it to bind to its host angiotensin converting enzyme receptor, which in itself is a carboxypeptidase. And if what's required is to induce strong virus neutralizing antibodies and T cell responses against that virus. And the point is there are lots of ways to do this. And I like Tony's slide, Dr. Fauci's slide, because he included ours, which is very generous of him. So, there are recombinant protein approaches, nanoparticle approaches, live attenuated viruses, adenovirus vector, genetic immunization, DNA and RNA vaccines, but they all more or less work by the same principles of inducing immune responses to the spike proteins. This is a little bit more detail coming out of a very nice review from Nature, where they describe in a little more depth the different technologies. So for instance, you have non-replicating virus vectors, such as adenovirus, which is involved in the Johnson & Johnson, the J&J vaccine licensed out of Harvard Medical School and Dan Baruch's lab. Also, the AstraZeneca-Oxford vaccine works by the same approach, as does the Russian adenovirus vaccine and one of the Chinese vaccines out of CanSino Bio. And then you have replicating virus vectors. These will come along a little bit later. For instance, Merck & Company has licensed 2 vaccines, one that uses the same technology as the Ebola vaccine, the vesicular stomatitis virus, which was very successful in taking care of the Ebola epidemic in DR Congo in 2019, using that to produce coronavirus spike protein, as well as a measles vector. And then there's some older technologies like an inactivated virus vaccine. There's a couple of them, such as Bharat in India, and Sinovac in China has an inactivated virus vaccine approach that also actually looks quite promising in terms of inducing neutralizing antibodies against the spike protein. And there is even a group looking at a weakened attenuated virus, which includes Codagenics based in New York in collaboration with Serum Institute of India. And then we know about the DNA vaccine approaches. So Inovio is taking this approach where DNA actually gets electroporated and gets taken up into the cell in the nucleus and produces RNA. And then there are at least 2 RNA vaccines being looked at from Moderna and Pfizer, where it's taken up by the cell, but the mRNA does not go into the nucleus. It gets produced into protein within the cytoplasm of the cell, and then those proteins and peptides are expressed. There are several different recombinant protein vaccine approaches, including ours, in collaboration with Biological E, and Sanofi Pasteur is pursuing a very promising protein subunit approach. And then there are virus-like particles, which are known as VLPs. An example of that would be the Novavax vaccine, which is also now in clinical trials. And the hope is that these vaccines will meet some of the following target product profiles. One, you want them to reduce the severity of illness. That would be really important to prevent you from going to the hospital. But also, ideally, they would also not only reduce severity of illness, but actually prevent you from getting the infection itself. And the reason that's important is if enough people get vaccinated with a vaccine that prevents infection, then you could interrupt transmission with sufficient herd immunity. And that's what happens with the measles virus. So we don't really know with these vaccines now in various stages of clinical development, whether they'll be able to meet all of these three criteria. Some may simply reduce severity of illness, not actually prevent the infection. Others may do that. So far, most of the vaccines have required 2 doses. They're to induce sufficient levels of virus neutralizing antibody and T cell responses. However, there's a new one that's now in clinical trials from J&J, the Ad26 vaccine, that there's going to be an effort to see if that will work with a single dose. And in the US, our program is known as Operation Warp Speed. It's a public-private partnership between the pharma companies and the United States government. And we're seeing there are at least 7 vaccines that'll be part of Operation Warp Speed. And the Warp Speed part is not so much compressing the clinical trials, but rather combining some of them and then producing vaccines, manufacturing at scale, even before we know the vaccines actually work, so that once we know they do, they're ready to go. And we're going to see multiple vaccines over the next year and determine whether they're actually safe and effective as they move through phase 3 clinical trials. The first wave we'll know by the end of the year whether either of these 2 mRNA vaccines out of Moderna and Pfizer are looking successful, as well as one of the adenovirus, the chimp adenovirus vaccine out of AstraZeneca, Oxford. And then we'll see more to follow: the protein particle vaccine from Novavax, the J&J vaccine, a Sanofi recombinant protein vaccine made in insect cells, and a Merck live vesicular stomatitis virus. So it's pretty exciting in that there's a diverse array of technologies, a high level of innovation, and the idea is we can have multiple shots on goal at producing these vaccines. And the fact that we're manufacturing a risk is a key part of accelerating it. So it's really important to, when we message about Operation Warp Speed, to say that the safety and the integrity of the clinical trials are not being compromised in any way. It's all of the pieces around it. Having said that, there are some potential disadvantages of the Operation Warp Speed program. No program's going to be perfect. There's a pretty heavy emphasis on the innovation, but some of these technologies, such as the mRNA and the recombinant adenovirus platforms, have never been licensed before. So some have suggested maybe we could balance it a bit more with some of the old school vaccines, like an activated virus or fermentation recombinant protein vaccines. It's quite an expensive program because of the manufacturing at risk. My only 2 real criticisms of the program are, I don't think communication is as strong as I would like it to be, given the magnitude of the program. You know, the emphasis has been around putting the companies in the lead. And so far we've seen a lot of missteps coming out of the companies from the way the CEOs and the boards have handled how they're doing this. And I really think it's going to be important for Operation Warp Speed to mount their own communication plan. And because we do have a very aggressive anti-vaccine movement in this country, and I'll talk more about that in a minute. My other issue is that we have this kind of U.S. go-it-alone strategy, because the U.S. government is now pulled out of the COVAX sharing mechanism and has pulled out of the World Health Organization. And we have this new concept that we've never had before, which I abhor. It's called vaccinationalism. You know, when we talk about vaccines now, we're talking about the Russian vaccine, the Chinese vaccine, the American vaccine, the British vaccine. we've never spoken that way about vaccines. It's always been about international cooperation. And I think it's going to slow the progress of getting COVID-19 vaccines out there. In the past, we've always focused on this in modern history, what I call vaccine diplomacy, which is, and the best example of that is when Albert Sabin developed the oral polio vaccine. Many people don't realize he didn't do it by himself, but he did it jointly with the Soviets at the height of the Cold War. So what happened was Sabin, who developed the oral polio virus vaccine strains, got back channel permission by U.S. State Department and the Soviet counterparts to bring the virus strains to the USSR, and they were produced at scale and tested on 10 million Soviet school children, shown to be safe and effective, and ultimately every Soviet citizen under the age of 20. And that's what led to the licensure of the oral polio vaccine, really critically important information. And then this was done again with smallpox. The Soviets had figured out how to freeze dry the vaccine. In fact, they called it dry vax, and cooperated with US scientists and D.A. Henderson to eradicate smallpox globally. And so I think we have to get back to that and somehow figure out a way to fix this concept of vaccinationalism, because I think it's going to interfere with our progress. The other thing I'm worried about is how much of Operation Warp Speed will filter to this problem, which is the large urban megacities that are now being affected by COVID-19 in low and middle income countries in Latin America, India, and Africa. We've heard about the corpses piling in the streets in Ecuador and Guayaquil this summer, and we're having similar problems in Belém and Fortaleza and Manaus in the Western Hemisphere. And of course, I'm worried about Lagos and Nigeria and the big Indian urban areas. I mean, how do you do social distancing under these kinds of circumstances? So while Operation Warp Speed is underway, there's a very robust program now happening through these organizations. They actually call themselves the Developing Country Vaccine Manufacturers Network. And there's some pretty interesting vaccines that are being developed jointly through by the DCVM members or through South-South partnerships. So for instance, Sinovac has an inactivated virus vaccine on alum that's being done in partnership with the Institute of Butantan in Sao Paulo. They're the public sector vaccine manufacturer in Sao Paulo. AstraZeneca Oxford is partnering with Fiocruz, which is like Brazil's NIH and CDC kind of rolled into one through their manufacturing arm known as Bia Manginos. In Indonesia, Sinovac is partnering with Bio Farma on an inactivated virus vaccine. And then in India, you have several different strategies. Bharat's pursuing an inactivated virus. The Serum Institute, which is one of the world's largest producer of vaccines, producing a lot of the vaccines for Africa, based in Pune in India, is partnering with AstraZeneca Oxford. And at Baylor, we're partnering both with PATH and this organization, Biological E, which is one of the big Indian producers, to scale up production of a recombinant protein corresponding to the receptor binding domain of the SARS-2 coronavirus. And the BioE is also partnering with J&J for an adenovirus vaccine. So there's a lot of stuff going on now outside of some of the US involved activities. And that's both interesting, but I'm also a little bit concerned that we're not seeing greater U.S. involvement on this international front. So in our case, our Texas Children's Center for Vaccine Development is partnering at Baylor College of Medicine. Their School of Tropical Medicine has been partnering with PATH, which is a nonprofit product development partnership based in Seattle, and they led the development of the meningococcal A vaccine for Africa, as well as the malaria vaccine for Africa, partnership with GSK, and now they're working with us. So our Center for Vaccine Development is historically focused a lot on parasitic disease vaccines, with a lot of support from NIAID and NIH. We have a schistosomiasis vaccine now and moving into phase 2 trials as for hookworm in Africa and Brazil, a new river blindness vaccine with the New York Blood Center and other organizations, a leishmaniasis vaccine with NIAID and Uniform Services, University and a Chagas vaccine with a consortium of Mexican institutions supported by the Carlos Slim Foundation, Clayburgh Foundation, and SWIMRI. And this is an initiative that's led by myself together with my science partner, co-partner for 20 years, Mary Elena Patazzi, focusing on recombinant low-cost global health vaccines. Now, we're not virologists, but about a decade ago, we were approached by the New York Blood Center, Shibu Jiang and Lan Yingdu, who had identified a very promising strategy for SARS-1 and MERS, and it was based on the receptor binding domain of the spike protein. So this is a cartoon of the coronavirus again. This is the trimeric spike protein. And then this is the receptor binding domain. They had found that if they produced that, they could immunize laboratory animals, such as a transgenic mouse model that was developed by Ken Chang at Galveston National Laboratory, a transgenic mouse with the ACE2 receptor, the angiotensin converting enzyme 2 receptor. And they could get high levels of protection without the immune enhancement that you sometimes hear about, where you get the paradoxical infiltration of eosinophils in the host tissues, which we attribute to a Th17 mechanism. And unfortunately, the time frame doesn't allow me to go into much detail about that, but we got very excited by that. We applied to the NIH and got support to develop the SARS-1 and MERS receptor binding domain. With our program officer supporting us, Eric Stemme. And we've been developing low-cost COVID-19 vaccines in this yeast Pichia pastoris system. And you might say, well, what do I care about Pichia pastoris? Well, it's a yeast, it's a methanol-utilizing yeast, and Pichia together with Hensenula, or some people call it Hensenula, are yeast that are used to make the recombinant hepatitis B vaccine locally in Bangladesh, Brazil, Cuba, India, Indonesia. Here's a scanning EM of those budding yeast. And we've been using that approach because this could afford an opportunity to start producing vaccine locally. So we manufactured the SARS-1 receptor binding domain at Walter Reed back in 2016. And now we've used that same approach and some bridge funding from NIAID to produce the SARS-2 receptor binding domain. And this has now been technology transferred to Biological E in India to produce 1 billion doses. And we've been able to show really exciting high levels of virus neutralizing antibody in animal models. And now this is being scaled in India. So we're hoping that we can come in on the global health front as well, and really getting around this problem of immune enhancement as well. Now, the other piece to this, of course, is the other rocky area is the fact that we've got a pretty aggressive anti-vaccine movement here in the United States. And as many of you know, I've been going up against this for years because I have a daughter with autism and intellectual disabilities, and I've been explaining why there's no link between vaccines and autism. The other problem, though, is the anti-vaccine lobby in the U.S. also, besides claiming that vaccines cause autism and other chronic illnesses, they say vaccines are rushed, they're not adequately tested for safety. There's conspiratorial conflicts between the pharma companies and the U.S. government, or this is conspiratorial collaboration. None of these things are true, but it's out there, and the problem is, without having that communication or plan around Operation Warp Speed, leaving it to the pharma companies, it's actually enabled the anti-vaccine movement. The other thing they do is they've put out, since 2015, they've been putting out this fake concept called health freedom or medical freedom that's been coming out of some far right-wing extremist groups in Texas, where I live and work, and elsewhere. And the consequence of that is we've had this devastating rise in the number of parents who are denying access to their kids for vaccines, especially around in the Austin, Texas area, around 72,000 kids not being vaccinated, according to our Texas Health Department, and summarized by the Immunization Partnership, which is a nonprofit here in Texas. And this doesn't even account for any of the 300,000 homeschooled kids. So we have a real crisis in Texas now because kids are being denied access to vaccinations. And then we have this other additional issue that now the health freedom guys are not only protesting against vaccine, but they've glommed on campaigns against social distancing, contact tracing, and masks. So it's a full-blown anti-science movement. And then in addition, we still have their assertions that vaccines cause autism and other chronic illnesses. And I've been up against that as well because I'm a vaccine scientist and the father of Rachel, who's I have 4 adult kids, including Rachel, has autism and intellectual disabilities, and wrote this book, Vaccines Do Not Cause Rachel's Autism, where I do a deep dive explaining why there's no link between MMR or thimerosal containing vaccines and autism and all the other things that they allege. And then also go into what autism is and how we've identified more than 100 genes involved in early fetal brain development. This is a major paper produced now at the Broad Institute at Harvard and MIT, which has identified these 100 genes involved in autism spectrum disorder expressed in the cortex of the brain during early fetal brain development. Most are expressed and enriched in excitatory inhibitory neuronal lineages, and many are involved in the neuronal cytoskeleton. So we did whole exome sequencing on Rachel and my wife Ann and I, and we've identified one of those genes. It's well, actually, it's not the same as the list of the 100, but it's related. It's a neural cytoskeleton gene encoding a non-red cell spectrum, a neuronal spectrum. The point is we have a very potent alternative narrative on what autism is. The problem, of course, though, is that this anti-science movement that started in the US is now globalizing. We have the Russians now filling our social media with anti-vaccine postings, and that's creating a huge, through bots and trolls, that's creating a huge problem. And the leadership of the anti-vaccine movement now was recently in Germany, speaking in a rally in Berlin with reports from CBS News and elsewhere that this was sponsored by far-right wing groups, including a neo-Nazi party in Germany. So I'm very worried about this globalization of American anti-science, and I think that's something we're really going to have to be concerned about. So I'll stop there to say I think I see us involved in this epic struggle between vaccines and vaccination and all of these new 21st century forces. This is the new book that will come early next year, Preventing the Next Pandemic, Vaccine Diplomacy and a Time of Anti-Science. And the point is the kinds of things that are opposing us are not things we ordinarily get trained to think about as scientists or physician scientists: poverty or climate change or war, political collapse, urbanization, or anti-science. But I think we're going to have to start thinking about ways to bring some of these things in, because they're doing a lot of damage and slowing us down. And that's why I call it the epic struggle. So thank you so much again. And I know there's people listening from NIH and NIAID, of course, and from the FDA and the other health and human services agencies. And I just want to take this opportunity to thank you again for your extraordinary heroism and helping us through this terrible COVID-19 pandemic and epidemic in the United States. So thank you very much.
Thank you, Dr. Hotez in absentia. Our second speaker of this session will be Dr. Julie Ledgerwood. Julie is the Deputy Director of the Vaccine Research Center in NIAID. She's also the Chief Medical Officer and Chief of the Clinical Trials Program within the Vaccine Research Center. Julie is going to talk to us about an update on the status of current vaccine clinical trials. Julie?
Yes, thank you, Rick. Can you hear me okay? Okay, perfect. So that was a nice overview, really a lot of information in Peter's talk, which is great. And so I'll just dive right into the point of my talk today is to tell you more about the vaccines that are in clinical development, with a real focus on those that are being developed by Operation Warp Speed, which Peter spoke of. So the premise behind Operation Warp Speed and the work I'll talk about today is really summarized nicely in this science paper I'm showing you the title page from here. And it talks about the importance of the need for a public-private partnership that could harmonize efforts across the country and work towards a rapid development of a COVID vaccine. So this is sort of the premise behind it. that there are really three entities related to that, or that came from that concept. One is Operation Warp Speed, which Peter mentioned, and most of you have probably heard of, and I've been a part of now for many months, and have really come to recognize that it's probably it's really history making, and I wish people understood it better. And so I'll try to help you understand it today in relation to vaccine development. There's also the active partnership with which Chris Austin and I think Tony mentioned this morning. And then the COVID network, which is, or the COVID prevention network, which is a cumulative network that brought together many, if not all, NIH-funded networks that have the ability to do trials, and DoD networks and academic networks that hadn't necessarily been part of either of those. And we'll talk about how they've been integrated into these efforts as well. So you just heard from Peter about some of these concepts, so I'll tell you now a little more. The Operation Warp Speed vaccine development program currently is moving forward with 6 vaccines. Additional vaccine or two may come along, but these are the vaccines in trials currently, and there are really 3 platforms. And so when you think about a vaccine, there's the platform or type of vaccine, and then there's the antigen or what is it immunizing against. So in all cases of these vaccines, as Peter mentioned, we are vaccinating to induce an immune response to the spike protein of SARS-CoV-2. And there are 3 main categories or platforms of vaccines that we're developing right now. Those are the mRNA from Moderna and Pfizer, the adenovirus vectors from AstraZeneca and Janssen or J&J. And I'll just say that these adenovirus vectors are quite different from one another. Not only are they different types of adenoviruses, they come from different families of adenoviruses, and they encode slightly different sequences of the spike protein. And then there are recombinant protein or nanoparticle-based protein vaccines, if you lump that into one platform, by Novavax and Sanofi, with an adjuvant, a different adjuvant for each company. So I'll talk about these today. The first COVID vaccine trial was launched in March, which you heard of, I'm sure. It's the Moderna mRNA product that was co-developed by NIAID and Moderna. And then Operation Warp Speed was officially announced in May, but the activities that led to it, the HHS funding of many of these programs, preceded that. And at this point, there have been a number of trials looking at the vaccines listed here in phase 1 and phase 2, and those have shown safety and immunogenicity. And there have been many animal studies looking at various animal models, including non-human primates, to show protection of the airway against SARS-CoV-2 challenges. And so all of the vaccines I'm showing you here have met the hurdles from those various checkpoints to continue to move forward in the Operation Warp Speed development umbrella. So starting with the mRNA vaccine, the Pfizer and the Moderna products are both mRNA vaccines encoding stabilized spike antigen. So there's a wild type spike option or a stabilized spike option, which includes a mutation in the amino acids to make it more stable and immunogenic. They both encode for that stabilized spike. So Peter already spoke about how mRNA is injected, and then the antigen of interest is translated in the cytoplasm, and then the immune response occurs against that. There haven't been licensed mRNA vaccines to date. There have been many types of gene-based, including mRNA vaccines in clinical trials, but not licensed yet. So he's right about that. However, there are they are advantageous because they are a platform-based technology that can be made very efficiently. So once you know the sequence of the antigen you want to vaccinate against, and you have a platform-based or process where you can make an mRNA vaccine, you essentially can plug in the antigen gene sequence of interest and make a new vaccine. So what NIAID was able to do with Moderna this year was to launch the development of this vaccine in early January, after the sequence of SARS-CoV-2 was shared publicly, and start that trial about 6 weeks later. So this was certainly record time, but the reasons were really a few. There are really a few reasons why we were able to do that. One was that SARS vaccines for SARS-CoV-1 and MERS vaccines had been in humans and had been safe. So similar antigens for spike protein had been safe in humans. mRNA vaccines of the same platform with different antigens have been in humans and were safe. And so we were able to move quickly without a lot of toxicology testing, and that's been done before. This wasn't novel conceptually. And so we did that. We launched the program quickly, and the product was safe and immunogenic and reported out in the New England Journal in July. Some of that data is here. And so I'll show you data from the trials that have been published for the platforms I'm talking about today. But the real take-home message is that they are all immunogenic in a range of similar to convalescent antibody response. So in all cases, the companies have assessed their immune response either by ELISA-based spike antigen assays or neutralizing antibody assays, or both, and compared that to convalescent serum pooled samples, a collection of samples of a variety of severities of illness, a variety of ages of recovered subjects or patients. And so here you can see the 25, 100, and 250 microgram dose. It was given twice on day 0 and 28, compared to convalescent samples, which are shown on the far right of the graph A. And so the point here is that it's similar. These titers are very high and reflect a dose that is protective in non-human primates. These are just two different assays I'm showing you here. One is the plaque reduction newt and one is an ELISA. So next mRNA vaccine that has been published for phase 1 results is the Pfizer product. And they're similarly, they have, this is the neutralization assay that they've chosen to use against comparing their vaccine responses against hemoconvalescent serum, which is on the far right of the graph again here. And you can see that depending on dose, they can overcome, even be higher than the convalescent serum titers, which we've seen with many of these candidates. So I'll just say in the case of the Moderna product, 100 micrograms is going forward. In the case of this Pfizer product, 30 micrograms is going forward. So they use the phase 1 data to select the dose that moves ahead, along with challenge studies in animals. So next, I'll quickly tell you about the ad vector or adenovirus vector-based vaccines. Adenovirus vector vaccines for humans have also not been licensed. Viral vector vaccines have, but not this particular type. And so these are somewhat novel. However, they've been studied in humans for almost 20 years or maybe more. Various types of adenoviruses have been studied carrying genes of various types in many different programs, including HIV and Ebola and flu. And so this is not novel technology, it's just new for the licensed vaccine world. So the way that this works is the adenovirus shell, which is replication incompetent, can't cause an adenovirus infection, is injected carrying the gene or the spike insert. And here is where there is a difference. So in these two vaccines, the Janssen and the Oxford or Oxford/AstraZeneca vaccine, they use a different type of adenovirus, and they use a different SARS spike antigen sequence, so they're slightly different. So first, the Oxford data came out describing their chimp or chimpanzee novel platform in a phase 1/2 trial. So this was in over 1,000 people, and they've shown it to be safe in that population, 18 to 55 years. And it was also immunogenic, so similar types of data here, showing either ELISA on the left or neutralization assays on the right, compared to convalescent samples. Again, you see that the vaccine, either given once or twice, can induce immune responses similar to those in recovered patients. And here the 5 times 10 to the 10th dose has gone forward into phase 3 trials. Next, the more traditional approach, or protein subunit vaccine with adjuvant, is being taken forward by Novavax and Sanofi GSK. The reason it says Sanofi/GSK is Sanofi is making the protein subunit, and GSK is likely making the adjuvant for them. So it's a combined effort. Novavax makes their own novel adjuvant to accompany their protein subunit. So far, the Novavax trial has published their phase 1 data, and they have numerous trials occurring right now around the world. Again, this product has also been shown safe, well tolerated, and highly immunogenic, even at their lowest dose, 5 microgram with adjuvant, and that's the dose that's being taken forward. Again, here shown the neutralization on the top and the ELISA on the bottom compared to human convalescent plasma. So similar kinds of demonstration of immunogenicity. And in all cases, these vaccines have shown at least some, if not considerable, protection in non-human primate models. So taking these candidates quickly from these phase 1 trials into large efficacy trials requires a lot of effort. And so Operation Warp Speed and each company have assembled teams of hundreds of people. So we call it an all of US government response. So there are DoD team members and HHS team members and beyond. And so it's really a broad effort. A lot of people have essentially detailed from their day job, if you will, to work on this full time. And I mean full time 7 days a week, around the clock sometimes. So it's really Operation Warp Speed just means many more people working, many more hours simultaneously, taking many steps in parallel. So oftentimes vaccines are developed over a period of 5 to 10 years, for example, and each step is taken in series. Here, everything is being done in parallel, as Peter mentioned. So the animal studies, the phase 1 studies, the launch of phase 3, the large-scale manufacturing, the plan for deployment, all being prepared in parallel with checkpoints along the way. At any point in time, if the vaccine doesn't meet the criteria, it would be dropped. So all these efforts are being done in parallel. So we decided to move forward with these efficacy trials by harmonizing efforts across the different companies. And that means that we told them, working with FDA and FDA guidance, that they needed to have a harmonized endpoint, which was symptomatic, virologically confirmed COVID-19. They needed to work with NIH networks, and they needed to use harmonized laboratory assessment so we could compare vaccines. So currently in phase 1, each company uses their own assay type. We wanted to harmonize that so we could compare vaccines more readily. The Data Safety Monitoring Board for these vaccine trials has been assembled by NIAID, and therefore is independent of all of these Operation Warp Speed trial companies. So the trials, the DSMB oversees are the Moderna, the AstraZeneca, the Janssen, the Novavax, and they will oversee the Sanofi. The Pfizer program is Operation Warp Speed involved in terms of manufacturing, but their trial is independent of our efforts. So they have an independent data safety monitoring board. So, and then there will be an extensive between trial statistical analysis for correlates of protection that's being arranged by NIH. So this may be very redundant to many of you, but I find that when I talk about this, it's really important to mention basic concepts. So we're talking about randomized placebo-controlled phase 3 COVID disease vaccine efficacy trials. And so the way I explain that is large numbers of people are needed, 30 to 60,000 per trial, to get 100 to 200 endpoints. And an endpoint means that someone developed COVID after completion of the vaccine regimen, a certain point in time in the study, and that it was symptomatic and it was diagnosed by the study team through a central laboratory mechanism. So there's a very defined set of criteria that define an endpoint. So additional COVID cases may occur in the trial, either early in the vaccine regimen or not be captured or not have symptoms. Those will all be captured in the data, but they don't contribute to the primary endpoint of 150 or so cases. So we need to vaccinate, monitor people closely. They don't know if they got vaccine or placebo. They go about their lives and they present to the clinical trial site for multiple vaccine assessments, including safety and immunogenicity. And at any point they have symptoms, they get tested. The definition of what is a symptom is also defined in each protocol, because as you can imagine, every sniffle doesn't require COVID testing, just as it doesn't in clinical care. So there's a defined set of symptoms for each protocol that define when a COVID test should be done at minimum. So what we're looking for is a reduction in the disease incidence in the vaccine group compared to the control group. And that at different times throughout the trial, the DSMB looks through these cases and evaluates whether or not we've met that target. And that's vaccine efficacy. These trials are, of course, all overseen and regulated by the FDA with this NIAID DSMB that I mentioned. You may wonder, asymptomatic COVID cases will also be defined by retrospective analysis looking at antibodies against nucleoprotein, which is a protein in the virus, but is not a protein in the vaccines. So any immune response to that protein then would have come from natural infection. So we can use that as a distinguishing feature for cases that occurred that we didn't pick up symptomatically. The FDA put out this very detailed and informative guidance this summer, and this was what was used to guide the phase 3 efficacy trial. So this came out before the first phase 3 trials were launched. This was published in June. The phase 3 trials, initial trials were launched in July, utilizing aspects of this. So they were all designed following these guidance, these principles in this guidance document. And what is that? Well, the highlights of that are that the trials would be randomized, placebo-controlled efficacy trials, as I explained. Sample size, to get enough primary endpoints, you need to have a sample size of 30 to 60,000 people per trial. Most trials have between 80 and 110 sites. They all utilize US sites, and some utilize international sites. We're looking at adults. We don't have any data yet in children to warrant including them at this point. We need safety data, and some would argue efficacy data in adults to put children into trials. Those are important next steps that are being considered right now with the FDA. And I know that at least Oxford has moved into adolescent or pediatric trials in the UK, I believe. And some of the international vaccine efforts have also moved into pediatric trials. Those will be important next steps with some of these products I've just mentioned. Primary endpoint we talked about, biologically confirmed symptomatic disease. The difference between the vaccine group and the placebo group, that's really important when you design a trial and you determine how many volunteers you need and how many cases you need to define your endpoint having been met. So the FDA said we want to see efficacy with a point estimate of at least 60%. And now when there's a point estimate, there's a confidence interval. We want to see that the confidence interval, the lower bound of that, is greater than 30%. So we are comfortable and confident that efficacy signal is true and real. There are a lot of reasons why that's important, but obviously most importantly for public health. The assays have been harmonized, correlates analysis across all trials, common DSMB I mentioned. So the status of the trials now, the Moderna mRNA and the Pfizer mRNA trials are the furthest along in terms of enrolling. I think the mRNA Moderna trial, they've enrolled over 27,000, and the Pfizer trial has enrolled over 30,000. They're both still enrolling, targeting primary endpoint cases in the 150 to 160 range. The AstraZeneca or Oxford, Chimpata Oxford trial, and the Janssen or J&J trial with AD26, they launched later, so they aren't as far along in the enrollment process, but they are ongoing as well. The Novavax and Sanofi trials are scheduled to launch later this year. I mentioned that there was a coronavirus prevention network that was stood up by NIH, and this is just describing how that worked out. So many DoD and NIH-funded networks came together to form this overarching coronavirus prevention network we call CoVPN. This is just a snapshot of sites for the initiation of CoVPN, but since then, large numbers of sites have been incorporated into the network. To identify where a trial should put their sites if they're trying to find endpoints quickly, we launched a predictive analytics working group within Operation Warp Speed, and I'm part of that. And so I've been a data analyst now for a total of about 4 months. So it's been fascinating to work with these real artificial intelligence and data masters in the private and public sector. So we work closely with CDC and others in HHS, and also with essentially two private industry groups, Palantir and Data Robot. And they are helping us to track the outbreak and to inform the sponsors, the companies, where we think the cases will be highest when they are going to be looking for primary endpoints. So if you are vaccinating with 2 shots and you need primary endpoints in November through January, you don't really want to know where cases are highest right now. You want to know where we think they will be the highest in that part of the calendar. So we use 2 different modeling approaches. One is the CDC ensemble model. That means is the CDC has taken 12 or 15, I think it is, private sector, academic sector models, and they've essentially combined them into the best model we could have that forecasts out about a month. When you get beyond a month, you lose specificity, sensitivity. But this model has been well validated compared to, so we do retrospective look backs and see how well it predicted, and it's very good. We use it primarily to show what's going to happen in the next month or what we think will happen. We use real data from USA Facts as the source, and the dashboard we use is called HHS Protects. Obviously, it's an HHS dashboard. It informs a lot of efforts in the government, but certainly Operation Warp Speed and our trial efforts. So this is a map showing at one snapshot in time, if we were to tell the system, predict the high number of cases per week on October 10th, where would they be by county? And this is the snapshot of that. And we can zoom in on these counties, we can put this in tabular form, and it helps us to decide where we should be looking for cases. It informs all of the trials, prevention or treatment, where they should have sites and so forth. We also incorporated large numbers of sites into the dashboard that we could view alongside or with, superimposed with the case and prediction data. And so it's hard to see because so many of them are in the same regions, but there are almost 1,000 sites in the dashboard now. These are private sector, academic, NIH funded, DoD, all kinds of sites have said, I want to be part of this, put me on the list, and if it makes sense, trial sponsors can contact us. So that's how it's working. And so all these different groups are involved in one or more trials at this point. It's really a broad effort. We can also zoom into these areas around a site or a given county, and we've incorporated in about 15,000 essential facilities into this dashboard. So we can see where all the Amazon warehouses are, meat packing plants, distribution centers for UPS, et cetera. Because those may, we don't know for sure, but they could, based on history, be the source of a micro outbreak. A micro outbreak meaning at a meatpacking plant, and that could turn into a community outbreak. So we have all of that kind of data we can think of into the dashboard. This is also helping to select sites. Where will we potentially find the most cases? So then in addition, we need to communicate the plan and what I've just told you. And it's really difficult to do that while working so quickly to launch these trials. And so I respect Peter's concern about the need for more communication, and we are making a lot of effort in that area, but recognizing that we can always do better, and we are. The community engagement efforts being launched by the NIH-funded networks are pretty intense, and they're coming forward now. So they've taken a while to get off the ground, but they're out there. This is an example. This is the Coronavirus Prevention Network website. If you look into this website, you can see an option to, you can see what trials are being enrolled, and you can see an option to register through a survey to be contacted by a site near you if you meet certain criteria based on the answers you give. So far, we've had almost 7 million views of the website, and almost 500,000 people have completed the registry and answered questions. It doesn't mean all of them are eligible for a trial or near a site, but that's how many people in the country have said, I want to be in a trial if I meet the criteria. So this has been key to helping our sponsors and their sites fill the trials. So I'll close there and just acknowledge all the partners involved by organization, and it's been a huge effort. Thank you.
Thanks very much, Julie, for that very nice overview of the current state of vaccine trials. Before we move on to our third talk, Julie, there's a couple of questions about vaccine-related questions in the Q&A at the end of the session. I'll probably ask you to address those. But our third and final talk is by Dr. Cliff Lane, who's the Director of the Division of Clinical Research within NIAID, and also the Deputy Director for Clinical Research and Special Projects within NIAID. And Cliff is going to review the current status of therapeutic trials and treatment guidelines for COVID-19. Cliff?
Thanks very much, Rick. So thanks very much. Happy to have a chance to review a few things today regarding treatments for patients with COVID-19. Just want to start by mentioning that even though there are products available under emergency use authorization and/or expanded access programs, there are currently no FDA-approved therapies for the treatment of COVID-19. And there are a very large number of clinical trials underway. In fact, as of the weekend, 2,233 studies for the treatment of COVID-19 were listed on clinicaltrials.gov. This is a graphic look at some of those studies that was put together by Stat News on July the 6th. And you can see by the size of the circle, the number of studies. And at that point in time, the agent getting the most attention was the combination of chloroquine or hydroxychloroquine. You can see 2 trials for which I think there was pretty clear evidence of efficacy. One of them, remdesivir. I'll talk about that more in a moment, and the other, dexamethasone. But the challenge is with all of this work going on, with so much information coming out, and I think Dr. Hotez mentioned some of the challenges with that, as did Dr. Cooper. It's really tough to have a sense of where you really have a new advance, where you have additional information, and trying to put all this into perspective is really tough. So one of the things that NIH has done is put together a panel to provide treatment guidelines for patients with COVID-19. I think Dr. Fauci mentioned this in his talk. This is indeed a living document. The guidelines panel meets multiple times a week and works to keep this document updated as new information comes in. It not only talks about the things that are put out by FDA under emergency use or expanded access, but it also talks about data on a variety of agents, many of which are available for off-label use, such as tocilizumab, or hydroxychloroquine, chloroquine. Some key recommendations from the current version of the guidelines panel that's up there is first, with regard to the use of chloroquine or hydroxychloroquine, the panel recommends against the use of these drugs for the treatment of COVID-19 in hospitalized patients. The guidelines panel has a rating system of A, B, C, A being something that the panel feels very strongly should be done. B is probably should be done, and then C is really optional, and then 1, 2, or 3 based upon whether or not the data are from randomized controlled trials, observational cohort studies, or a recommendation based just on expert opinion. So I've put here 3 A1 recommendations. The second, the panel recommends remdesivir for 5 days or until hospital discharge for patients who require supplemental oxygen, but not those who require high flow oxygen or mechanical ventilation. And I'll show some of the data supporting that recommendation in a moment. Third, the panel recommends dexamethasone 6 milligrams per day for up to 10 days or until hospital discharge for the treatment of COVID-19 in hospitalized patients who are mechanically ventilated. And then finally, another treatment that is available under emergency use authorization and quite popular, convalescent plasma. The panel has taken the position that there are insufficient data to recommend either for or against the use of convalescent plasma for the treatment of COVID-19. When one thinks about treatment of patients, I think it's helpful to look at these different disease manifestations, severity of disease, and think about it on a spectrum ranging from individuals with no symptoms, some of whom are probably out there helping to spread the infection, to outpatients with mild symptoms, inpatients not requiring oxygen, inpatients on low flow, high flow oxygen, and then finally mechanical ventilation. What I try to show you in some of the data from the remdesivir study and the dexamethasone study is that the antiviral interventions probably are going to have their biggest impact early in the course of infection, and some of the immunomodulatory interventions like corticosteroids are likely going to be of most value late in the course of the disease. So again, for the remainder of the presentation, I'd like to spend a few moments talking about remdesivir, dexamethasone, convalescent plasma in some detail, and then conclude with a look at some of the major NIH-sponsored clinical trials that are currently underway or planned to be underway in the very near future. So first, remdesivir. Remdesivir is a prodrug of an adenosine analog. So it's a nucleotide inhibitor. It's an RNA-dependent RNA polymerase inhibitor. It was initially studied fairly extensively as a potential treatment for Ebola virus disease, where it was not found to be of clinical benefit compared to 2 different monoclonal antibody regimens, but it did at least have a relatively good safety profile generated in humans during that study. It's given intravenously as a loading dose of 200 milligrams, followed by 100 milligrams IV daily for a total of 5 to 10 days. And data I will not go into. Gilead, the company that makes the remdesivir, has demonstrated relative comparability for 5 days versus 10 days. So hence the recommendations of the panel go with 5 days. As I mentioned, it has been studied pretty extensively for Ebola. It was also studied in a randomized controlled trial in China, which unfortunately was closed prior to reaching their desired sample size. And thus, while it has data consistent with the larger study that was done subsequently, it didn't have sufficient power to draw a clear conclusion. And again, it's been used quite extensively now to treat hospitalized patients in the US. The initial report on this study, known as Act 1, came out on May 22nd. John Beigel was the principal investigator. The bottom line finding, or the top line finding of this randomized controlled trial was that patients who had received remdesivir had a 32% faster time to recovery than those who received placebo. There was also a trend towards a survival benefit. The study enrolled 1,063 patients from 10 countries in the US, Europe, and Asia. Eligibility criteria included adults hospitalized with COVID-19 and some evidence of pulmonary disease. The randomization was standard of care plus placebo versus standard of care to remdesivir. The primary endpoint was defined as time to recovery using an ordinal scale, and I'll go over that in a moment. So it was basically time to ordinal scale 1, 2, or 3. The study opened February 21st and closed April 19th after achieving a total enrollment of 1,063. You can see here the distribution of sites across the US, South Korea, Japan, Mexico, Greece, the UK, Germany, Denmark, Spain, and Singapore. This shows the time to recovery for the remdesivir group on the top, the placebo group on the bottom. Again, recovery was defined as achieving ordinal scale 1, 2, or 3, namely not hospitalized at full activity, not hospitalized with limited activity, and hospitalized but for non-medical reasons. Patients were entered into the study from ordinal scale 4 to 7, hospitalized not requiring oxygen, hospitalized requiring oxygen, hospitalized requiring high flow oxygen or non-invasive ventilation, or hospitalized with mechanical ventilation or ECMO. So again, Individuals entered the study, ordinal scale 4 to 7. Time to getting to ordinal scale 1 to 3 was then time to recovery. You can see a splay after about five days, by the remdesivir group compared to the placebo group. If one looks at some of the subgroup analyses from this trial, you can see that there was a trend towards individuals of a younger age doing better, looking at the hazard ratios over on the right. The higher the number, the more the remdesivir arm did better compared to the control arm. There was very little difference seen, however, on basis of sex or duration of symptoms. There was an interesting trend in ordinal score with patients at lower ordinal score having a better treatment effect than patients at higher ordinal score. While each one of these subgroups was not individually powered to make a call, there was clearly a trend within these groups, with the earlier groups doing better than the more advanced groups. And that finding has been taken into account with regard to the treatment recommendations by the panel. Just looking at mortality over the entire study period, the hazard ratio for survival was 0.73, so basically a 27% improvement in survival for the patients in the remdesivir group. However, this did not reach a statistical significance of 0.05, so it's really referred to as a trend rather than the hard finding. Of note, though, the same trend to survival was seen as was seen with the trend towards time to recovery, with the best survival hazard ratio, best therapeutic effect from the perspective of survival being seen in those individuals entering the study in ordinal category 4, the hazard ratio of 0.82, ordinal category 5 with a hazard ratio of 0.3, and those patients coming in with more advanced disease, ordinal category 6 or 7, having a little, if any, or no evidence of a clear benefit on survival. Again, these groups are not independently powered. These should be viewed as trends as opposed to hard findings, but they do influence the treatment recommendations and are quite a bit in contrast from what I'll show in a moment with dexamethasone. So again, based upon these data and the other data provided by a variety of other studies, the guidelines panel has recommended remdesivir as a treatment for 5 days or until hospital discharge for patients who require supplemental oxygen, but not those who require high flow oxygen or mechanical ventilation. Indeed, those patients, may be given remdesivir. It's just not with the strongest recommendation from the panel. I'd like to make a few comments now on dexamethasone. Dexamethasone is obviously a glucocorticoid hormone with little mineralocorticoid effect, which is why it was chosen for study, in this case by the recovery group in the UK. Dexamethasone has been in medical use since 1961. It's a broad spectrum anti-inflammatory and a broad spectrum immunosuppressive. Some of its immunosuppressive activities are reflected in the fact that you can see exacerbations of viral infections like varicella zoster or parasitic infections like strongyloides. If one looks at the history of the use of dexamethasone in patients with severe viral respiratory infections, the data really have been conflicting. This study that was published in July by the Recovery Collaborative Group, however, really, I think, put to rest any controversy about whether or not dexamethasone is of value in patients with COVID-19 pneumonia and severe COVID-19 disease, and that they found that dexamethasone reduced 28-day mortality by 36% in ventilated patients and by 18% in other patients receiving oxygen. There was no benefit seen for patients hospitalized who were not receiving respiratory support. Again, this was a very large study by a trial that had a very, I think, valuable design, and that it was a real world randomized controlled trial facilitated by the fact that the UK has a national health system. The primary endpoint for this study was 28-day mortality. And what was done is this was set up as a computer system. So essentially any physician throughout the UK could randomize patients to this trial. It takes patients and it randomizes them to the standard of care or standard care plus any one of a number of arms. So the currently active arms are dexamethasone in children. As I mentioned, it's shown efficacy in adults, so there's no longer randomization to adults. Adults will be getting it as part of standard of care if they meet the criteria. Another randomization arm is azithromycin. Convalescent plasma is there as a factorial randomization with the above arms. And recently they've added the Regeneron monoclonal antibody as a additional arm. They have a second randomization to tocilizumab for patients with more advanced disease. The hydroxychloroquine arm was stopped, seeing no effect with mortality of 21% in the control versus 22% in the hydroxychloroquine. And the lopinavir, ritonavir, or Kaletra arm was also stopped for no effect with 24% mortality in the control arm versus 26% in the experimental. Here you can see the survival curves for the dexamethasone study by recovery, the top left being all participants with lower mortality rates in the dexamethasone group in red compared to usual care in blue. If you look over to the right, this effect was most pronounced in the patients on invasive mechanical ventilation with a rate ratio of 0.64. Bottom left, patients receiving oxygen only, a rate ratio of 0.82. And then interestingly, patients not receiving oxygen, a rate ratio of 1.19, while not statistically significant on its own, in that it crosses a rate ratio of 1. This, I think, is pretty strong evidence that, in fact, dexamethasone might, if anything, be harmful in this particular group. Other studies have been published looking at the role of systemic corticosteroids. A recently published prospective meta-analysis of 7 randomized trials, including 1,700 patients, noted that there was a 28-day mortality reduction of 34% in critically ill patients with COVID-19 who received corticosteroids. So taking these data together, the guidelines panel recommends dexamethasone 6 milligrams per day for up to 10 days or until hospital discharge for the treatment of COVID-19 in hospitalized patients who are mechanically ventilated, with the strongest recommendation and with a slightly weaker recommendation for patients who are hospitalized and receiving oxygen but not mechanically ventilated. So if one looks at the survival data from the recovery or the dexamethasone study and compares it to the survival data from the remdesivir or the ACT-1 study, what you find, again, looking at recovery rate ratios for dexamethasone, that the rate ratio is lowest and other enhanced survival for patients with mechanical ventilation. In contrast, if you look at the hazard ratio, sort of the equivalent measurement in ACT-1, you see a greater effect in patients with somewhat milder disease. So again, I think remdesivir appears to have its greatest effect earlier in patients who are hospitalized with and receiving oxygen, and dexamethasone appears to have its greatest effect in patients with more advanced disease. Single donor convalescent plasma is something that has been used since the earliest days of the outbreak, even in China. It is a perennial favorite in the setting of any new outbreak, dating back to the 1918 influenza pandemic. Even before that, it was used as a way of treating diphtheria and tetanus. There are multiple anecdotes in the literature of single donor plasma being of success in diseases ranging from influenza to SARS to Ebola. However, there are very limited randomized controlled trial data on this particular approach. Of note, a trial done in Argentina in 1970, reported in 1979, showed improved survival in patients with Argentine hemorrhagic fever, 16.5% mortality in the control arm versus 1.1% in the treatment arm. The plasma had to be given within 8 days of symptom onset, and there was a delayed neurologic syndrome, which appeared to be transient in those who received convalescent plasma. In 2019, a randomized control trial of high titer versus low titer anti-influenza A plasma failed to show any significant benefit. So again, we don't have a very strong evidence base for convalescent plasma, although it makes really good sense on a theoretic basis. So in COVID-19, this has been very widely available. It really has come into the clinic in 3 waves. The first of these being single patient use through emergency IND, while standard plasma is a licensed blood product. Immune plasma or convalescent plasma is actually viewed as an investigational blood product. So to use it, one needs to have approval through one or another mechanisms of the FDA. So again, single patient access via emergency IND became such a large enterprise for everyone involved that it was moved from emergency IND, single patient INDs to an expanded access program by the Mayo Clinic. Now, one of the requirements that you have ideally before going to an expanded access program is that you have randomized controlled trials in place set to give you an answer so that you know relatively soon whether or not the intervention is of value. And unfortunately, those trials really are still underway, so we don't have the definitive data at this moment. However, based upon an analysis, which I'll show in a moment, of the data from the expanded access program, an emergency use authorization has been issued. Just to note, this does not constitute drug approval. What it does, it facilitates the availability and unapproved uses of medical products during a public health emergency. Criteria include that when it's dealing with a serious or life-threatening condition, that there is sufficient evidence the product may be effective. So again, it's a relatively low bar for efficacy, that the potential benefits outweigh the potential risks. In the case of convalescent plasma or plasma in general, the risks are relatively well known. And finally, that there are no adequate approved alternatives. The Mayo Clinic Expanded Access Program was huge. It was an amazing undertaking, and the people involved did a great job. The study closed, the Expanded Access closed August 28th, moving everything over to emergency use. It involved 2,700 sites, close to 15,000 physicians, 105,000 patients, and 82,000 infusions. These are some of the exploratory efficacy data from that expanded access program. What was done was that patients were split into groups based upon the titer of plasma, the titer of antibodies in the plasma that they received. So the predefined analysis is shown over on the left, looking at outcomes for patients who were intubated in orange, patients who were intubated in blue versus patients who were not intubated in orange, splitting them into groups based upon whether or not they received plasma with a titer of greater than or equal to 1 to 320 or less than or equal to 1 to 160. So looking first at the blue lines for intubated patients, if anything, there was a slight increase in death rates, but not statistically significant, looking at low titer versus high titer. In contrast, for those who were not intubated, there was a statistically significant decrease in 7-day mortality. We don't have any data, at least that I'm aware of at the moment, regarding 28-day mortality for this group. In a post hoc analysis, splitting out patients who were non-intubated, treated within 72 hours of diagnosis, less than or equal to 80 years of age, who received high titer plasma greater than 1 to 320, there was a pretty marked decrease in 7-day mortality. Again, this was a post hoc analysis, and one is looking for additional confirmatory data to support this. And more importantly, I think the results of the ongoing randomized controlled trials. So in summary, there's a suggestion that convalescent plasma is an effective treatment for a subset of patients, in particular those less than 80, less than 3 days from diagnosis, with a plasma titer above, it was 1 to 640 or 1 to 320 by a neutralization assay that correlates with about 12 ortho units in the ortho assay. Convalescent plasma is currently widely available in the US and elsewhere, but in the US, it's under an emergency use authorization. There are no compelling randomized controlled data yet available. There are trials ongoing. The guidelines recommendation is that there's insufficient data to recommend for or against, and I think these data do support studies of antibody therapy as a potential therapy for COVID-19. And as I mentioned, the current recommendation from the guidelines panel is that there are insufficient data to recommend either for or against the use of convalescent plasma for the treatment of COVID-19. And recently there's a brief opinion piece in the Annals of Internal Medicine published last week, sort of going through this in more detail. I'd like to conclude in the last few minutes talking about some select NIAID-supported clinical trials. As Julie mentioned, much of this work comes out of Operation Warp Speed and the accelerating COVID-19 therapeutic interventions and vaccines or active partnership. Among the studies are randomized controlled trials of convalescent plasma run by NCATS or NHLBI, and you heard Dr. Austin mention a little bit of that earlier today. A study of immune IVIG by NIAID, following the Act 1 remdesivir study, the Act 2 and Act 3 trials looking at baricitinib and interferon beta respectively, and then the active portfolio of studies 1 to 5 looking at immunomodulators, monoclonal antibodies, and anticoagulants. As was mentioned both by Julie and Peter, the Trump administration announced Operation Warp Speed in May as a national program to accelerate development, manufacturing, and distribution of COVID-19 vaccines, therapeutics, and diagnostics. It's a public-private partnership. Monsef Slaoui is the chief scientific advisor, and the chief operating officer is General Perna. At the NIH level, under Dr. Collins's leadership, there has been launched the Accelerating COVID-19 Therapeutic Interventions and Vaccines, or ACTIVE partnership, to try to standardize and share preclinical evaluation methods in an open forum, and Dr. Austin spoke about this a bit, to prioritize and accelerate the clinical evaluation of therapeutic candidates with near-term potential, to maximize clinical trial capacity and effectiveness, and to advance vaccine development by encouraging some of the harmonization that Julie had mentioned. A couple of the trials I'll then mention, Dr. Austin mentioned the randomized controlled trials of plasma. There are 66 interventional trials of convalescent plasma currently on ct.gov. One he didn't mention is the one by David Sullivan out of Johns Hopkins. It's an outpatient study. He did talk about the study, the Pass It On study out of Vanderbilt, supported by NIH, NCATS, and Dolly Parton as well. Todd Rice is the PI. This is an inpatient study or in the ER to be admitted, looking at an ordinal scale outcome at day 15. The Hopkins study is looking at hospitalization or death as a primary endpoint. The other two studies, both of which are NIH supported, is the one out of NYU, Dr. Ortegoza. This is a convalescent plasma to limit COVID-19 complications in hospitalized patients. This is enrolling patients hospitalized for less than or equal to 72 hours on supplemental oxygen, looking at plasma versus saline, ordinal scale at day 11. Then finally, the one that NHLBI is sponsoring out of the emergency room network, SIREN, looking at diagnosed cases in the ER with less than 7 days of symptoms, plasma versus saline, and death, admission, or follow emergency care within 15 days. We're hopeful that these studies will have some pretty clear data to support the use or indicate that the use is not of value, convalescent plasma. Hyperimmune immunoglobulin operates on the same theory, basically taking the antibodies from multiple units of convalescent plasma, concentrating it into a drug product. It gives you the uniformity that you don't have with convalescent plasma. And indeed, there are licensed products of hyperimmune immunoglobulin, including those to prevent post-transplant CMV disease and post-exposure prophylaxis of varicella zoster in high-risk individuals. Two approaches are being taken to look at an anti-SARS-CoV-2 IVIG, genetically modified cattle, and then a more traditional way, patients who've recovered from COVID-19. NIH, NIAID is involved in two randomized placebo-controlled trials that are being planned. The first of these, likely to start within the next couple of weeks, is an inpatient study in patients with less than or equal to 8 days of symptoms, randomized placebo or IVIG, all of the individuals receiving remdesivir. That will be followed by an outpatient study in patients at high risk of serious complications. The corporate partners in this include Emergent, Grifols, and the Plasma Alliance, which includes Takeda and CSL Behring. The adaptive COVID-19 treatment trials, or the ACT studies, are sponsored by the NIAID Division of Microbiology and Infectious Diseases under Dr. John Beigel. First of these I mentioned, which was Act 1 of placebo versus remdesivir, showing remdesivir superior to placebo. The Act 2 study, remdesivir versus remdesivir plus baricitinib, has completed enrollment. The data are currently being analyzed and submitted for publication. And the group has moved on, is now enrolling patients in the Act 3 trial, which is remdesivir versus remdesivir plus interferon beta. The active portfolio of studies, 1 to 5, are shown here. Active 1, sponsored by NCATS, is looking at 3 different immunomodulators in patients receiving a background of remdesivir. They're looking at abatacept, CTLA-4 IG, cenicriviroc blocking CCR2 and CCR5, and infliximab to block TNF. The Active 2 study is a collaboration with the Division of AIDS and NIAID as the sponsor. It is an adaptive trial platform where one can bring in different therapeutic interventions that may appear to be of value based on prior clinical phase 1 criteria. It is an adaptive trial, Active 2 in outpatients, Active 3 in inpatients. Currently, Active 2 is comparing placebo to the Lilly monoclonal antibody COV555 in outpatients. Active 3, again, is an adaptive trial in inpatients. Here, the patients have a background of remdesivir. It's currently enrolling patients in a trial comparing placebo to the same Lilly monoclonal antibody. Indeed, as additional agents come in, they will be able to join these 2 studies. I'll show a schematic of that in a moment. And then finally, Active 4, sponsored by NHLBI, is a series of 3 trials looking at different anticoagulation strategies, 1 in outpatients, 1 in inpatients, and then 1 in discharged patients. This is the design of the Active 3 trial. What it tries to do is, rather than conducting 5 different randomized controlled trials, it has multiple arms in a single randomized controlled trial, looking at what's called the stage 2 because of its analogy to a phase 2, and then stage 3 because of its analogy to a phase 3. So as a product is qualified to enter the trial, here treatment 1 to treatment 4, it enters the trial. There's concomitant control arm being enrolled. At a midpoint evaluation or an early point evaluation, those products that are looking promising continue on into stage 3. Those products that are not are dropped out, giving you a way to test multiple products more quickly than you could doing a variety of individual trials. Again, I'll just conclude by saying there is a lot going on in this particular area. I think one of the best ways, what we've been trying to provide a place that people can go to get updated information about not only what's going on with the studies, but which of the treatment regimens that are available are indeed recommended and what the strength of those recommendations are at this particular website. And Dr. Fauci had mentioned this as well. So http COVID-19 treatment guidelines.nih.gov. Thanks very much.
Thanks very much, Cliff, for that comprehensive overview. Because we're running a little late on time and we want to make sure that Dr. Emanuel's session starts at 4, I think we will skip the 10-minute break. And in terms of the Q&A, since all the questions thus far are related to vaccines, Dr. Ledgerwood has volunteered to read them quickly and provide responses. Julie?
Sure, thanks, Rick. So first question is, what about the VSV live recombinant vaccine? So I believe you're talking about the Merck vaccine in development, and that is just not as far along. And so I didn't include it in my summary, but it is in development. Then are there any SARS-CoV-2 vaccines delivered intranasally like flu mist? So there are. They're not part of the Operation Warp Speed portfolio. My understanding is they're not as far along. It's mostly done by international groups, I think predominantly in China. They're doing this look. I mean, it makes some sense. As you know, it's a respiratory transmission virus. And so taking that as a research look makes some sense, but we're not doing that in Operation Warp Speed at this time. Does the fact that commercially available serology tests detect nucleic capsid, but vaccines express spike, create an issue in terms of back to work school testing? So the emergency use authorization assays approved by the FDA for emergency use either detect antibodies to nucleic capsid or spike or both. So there's a variety. The vaccines should induce antibody to spike, not nucleic capsid. Wild type infection should induce antibodies to both. So depending what you're looking for and what you need to find, you know, there's a test that would help you do that if you understand the virology and immunology there. Okay, Dr. Cohen said, antibodies are benchmarked against convalescent sera. What about T cells? So all the vaccines in phase 1 had to demonstrate T cell responses. Most of them used intracellular cytokine staining and looked for antigen-induced T cells, so T cells specific for the spike antigen, and looked to see what kinds of cytokines they make. And what we're looking for here is mostly a safety signal. and meaning that we have a theoretical idea that a Th2 skewed cytokine profile in antigen-specific T cells might be a bad thing. We don't know that, but we looked for it, and so the vaccines going forward have a Th1 skewed response, either predominantly or entirely based on assays done to date. And so we, again, don't know if that means anything, but we've done that as an assessment, mostly for safety. It may be important for immunology and for immune response, but that part, the correlates aspect is still being worked out. When will the phase 3 trials have sufficient data? So as Dr. Fauci said this morning, we think that the trials that started in July might have some interim data by the end of the year to tell us if they're efficacious. The others would come along after that early next year, if all goes well. Does the experience of warp speed suggest that future pandemics follow a similar model, or should the U.S. identify a common backbone for the next emergent disease? I'm going to let Cliff answer that one.
So I don't think a common backbone necessarily will be right for every different infectious disease. So what I like about what Operation Warp Speed has done is really sort of de-risked the process for the companies so that multiple platforms are able to come in at the same time. I do think the notion of trying to have the U.S. government coordinated has been really helpful. I can say that one of the things that I had observed on a travel to China early in the outbreak was there was a lot of research going on, but there wasn't any priorities being set in any way as to which studies were most important, thus which studies would get done. So as a result, you had a ton of studies being initiated, but many of them, the vast majority of them, never getting to completion. So I think having a coordinated effort is really quite helpful. Thanks.
Thank you, Cliff. So with that, we will conclude this session and turn things over to Lisa for the final session. Lisa.